Cholesterol is the precursor of all steroid hormones, including corticosteroids and sex hormones. They are hydrophobic. They use carrier proteins for transport in the blood. Cortisol and corticosterone mainly use transporting. Aldosterone and progesterone mainly use albumin. Sex hormones use sex hormone binding globulin. Here, presence of few letters may help you remember corresponding steroid hormones each carrier protein transports. For example, COR reminds you of cortisol and corticosterone. AL reminds you aldosterone. Steroid hormones control gene transcription through nuclear receptors. For example, aldosterone can penetrate plasma membrane and enter the cell. It binds aldosterone receptor. The complex is translocated to nucleus where it binds DNA and controls transcription of target genes. Steroid hormones are produced by adrenal cortex, ovaries, and testes. Today, we will focus on adrenal cortex. Adrenal glands, also called suprarenal glands. Suprarenal means on top of the kidney. This is the kidney, and this is adrenal gland. Each adrenal gland contains two endocrine glands, adrenal cortex and adrenal medulla. Adrenal cortex produces aldosterone, cortisol, and androgens. Adrenal medulla secretes norepinephrine and epinephrine. These are unrelated chemicals, but all help with extreme situations. This is adrenal gland, this is adrenal cortex, and this is adrenal medulla. Adrenal cortex can be further divided into three layers. First layer is zona glomerulosa, followed by zona fasciculata and zona reticularis. Zona glomerulosa secretes aldosterone. Zona fasciculata and zona reticularis secrete cortisol and androgen. Adrenal medulla secretes epinephrine and norepinephrine. Today, we will cover two topics. First, we will look at corticosteroids. Corticosteroids include mineralocorticoids, glucocorticoids, and androgens. Aldosterone is the main mineralocorticoid. This is aldosterone. It plays a role in control of salt and water balances. Cortisol is the main glucocorticoid. This is cortisol. It plays a role in regulation of metabolism of glucose, lipids, and amino acids. This is angiostenedione. It is an androgen. Aldosterone is secreted by zona glomerulosa in response to a decline in either blood volume or blood pressure. Aldosterone functions through a nuclear receptor. It promotes distal and collecting tubules in the kidney to reabsorb sodium. Here is integration with renal physiology. After sodium is reabsorbed, water follows. Blood volume increases. As a result, blood pressure increases. This is a principal cell in the collecting duct. And this is the interstitial space, and this is the lumen. Aldosterone enters the cell binds the nuclear receptor. The complex controls transcription of several target genes. Among them, one gene encodes sodium-potassium ATPase. The other one codes for epithelial sodium channel, or INAC. INAC allows rapid diffusion of sodium into the cell. Sodium-potassium ATPase pumps 3 sodium ions out of the cell and brings 2 potassium ions into the cell. This creates a concentration gradient, which results in reabsorption of sodium and water into the blood and secretion of potassium ions into the lumen. Glucocorticoids are essential for life. Cortisol is important in resisting stress and inflammation. It helps the body deal with stressful situations caused by physical, psychological, and physiological stressors. Glucocorticoid receptors are found in most tissues. Cortisol regulates and supports a variety of important cardiovascular, metabolic, immunologic, and homeostatic functions. This is a hepatocyte. Cortisol enters the cell and binds nuclear receptor. The nuclear receptor is translocated to nucleus and binds DNA and controls transcription of several target genes. Among them, PEBCK codes for PEP carboxy kinase. In this case, cortisol receptor works together with CREB. CREB is controlled by glucagon. As a result, cortisol increases gluconeogenesis. Cortisol also causes a moderate decrease in the rate of glucose utilization by most cells in the body. Cortisol is a stress hormone. Stress hormones act to mobilize energy stores. For example, cortisol mobilizes amino acids from non-hepatic cells and increases blood amino acid levels. Cortisol promotes mobilization of fatty acids from edible tissues and enhances the oxidation of fatty acids in extrahepatic cells. As a result, cortisol forces extrahepatic cells to switch to fats as energy sources. This is a steroid hormone secreting cell. These are smooth ER, and these are mitochondria. Steroid hormones are synthesized in the mitochondria in smooth ER. Steroids directly diffuse across plasma membrane and therefore does not require exocytosis. For synthesis of steroid hormones, the first step is cleavage between C20 and C22 by desmolase. This reaction converts cholesterol to pregnenolone. It occurs in mitochondria. It is a rate-limiting step. It requires NADPH and oxygen. It is regulated by hormones. The second step is oxidation and isomerization of pregnenolone by 3-beta-hydroxysteroid dehydrogenase. From progesterone, two oxidation reactions at position 21 and 11, followed by oxidation at position 18, lead to aldosterone. From the same progesterone, three hydroxylation reactions at position 17, 21, and 11 leads to cortisol. These hydroxylation reactions are catalyzed by 17-alpha hydroxylase, 21-alpha hydroxylase, and 11-beta hydroxylase respectively. From progesterone, hydroxylation at position 17 followed by removal of carbon tail catalyzed by 1720 lyase leads to androstenedione. Hydroxylases that modify progesterone to other steroid hormones are mixed-function oxidases that require NADPH and oxygen. They are members of P450 superfamily. Deficiency of 3-beta-hydroxysteroid dehydrogenase results in absence of aldosterone, cortisol, and androgens. Absence of aldosterone causes hypotension. Absence of androgens causes female-like genitalia in males. Deficiency of 17-alpha hydroxylase results in absence of cortisol and androgens and overproduction of aldosterone. Overproduction of aldosterone causes hypertension. Absence of androgens causes female-like genitalia in males. Deficiency of 21-alpha hydroxylase results in absence of aldosterone and cortisol and overproduction of androgens. Absence of aldosterone causes hypotension. Overproduction of androgens leads to masculinization of external genitalia in females and early virilization in males. Deficiency of 11-beta hydroxylase results in increased production of 11-deoxycorticosterone and overproduction of androgens. Increased production of 11-deoxycorticosterone causes hypertension. Because this hormone also suppresses the Renin-Angiotensin system, it causes low renin hypertension. Overproduction of androgens leads to masculinization of external genitalia in females and early virilization in males. Here is regulation of glucocorticoids. The rate-limiting step for production of glucocorticoids, especially cortisol, is catalyzed by desmolase. The activity of this enzyme is stimulated by ACTH. In contrast, the rate-limiting step for production of aldosterone is catalyzed by aldosterone synthase. Activity of this enzyme is stimulated by angiotensin II, which we will discuss later. Desmolase, also known as cholesterol side chain cleavage enzyme, or P450-SCC. Here, SCC stands for side chain cleavage. P450-SCC is a mitochondrial enzyme that catalyzes the first reaction in the production of various steroid hormones. This enzyme is encoded by CYP11A1 gene. In cells from zona fasciculata of adrenal cortex, transcription of this gene is induced by ACTH. With stress or changes in circadian rhythm, hypothalamus secretes CRH, which acts on anterior pituitary gland to stimulate ACTH secretion. ACTH acts on adrenal cortex to stimulate secretion of glucocorticoids. Glucocorticoids increase gluconeogenesis in the liver, increase protein catabolism in the muscle, and lipolysis in the edible tissue. Glucocorticoids also suppress immune system. Glucocorticoids have a negative feedback loop to suppress release of CRH by the hypothalamus and ACTH by anterior pituitary gland. Here, hypothalamus secretes CRH. CRH is a 41 amino acid peptide hormone. Pituitary glands expresses CRH receptor. It is a G protein coupled receptor. Activation of CRH receptor by CRH activates transcription of ACTH through a cyclic AMP-dependent pathway. ACTH is a 39 amino acid peptide hormone. ACTH is synthesized from pre-pro-opium melanocortin or pre-POMC. Adrenal cortex expresses ACTH receptor, a G protein coupled receptor. Activation of the receptor by ACTH activates transcription of CYP11A1 gene coding for desmosterase through a cyclic AMP-dependent pathway. Anterior pituitary glands synthesize pre-POMC. Removal of signal peptide leads to POMC. This is POMC. This protein undergoes a serious post-translational modification before it is cleaved by endopeptidase PC1 and PC2. PC1 is present in anterior pituitary gland. PC2 is produced by hypothalamus. Cleavage of POMC by PC1 yields ACTH among other polypeptides. Cleavage of ACTH yields alpha melanocyte stimulating hormone. This hormone is the most important melanotropin for skin pigmentation. It is derived from ACTH. Because ACTH contains an alpha melanocyte stimulating hormone sequence, it has about 1/30 as much melanocyte stimulating effect as melanotropin. Because the quantity of melanotropin are very low in humans. In contrast, ACTH levels are quite high. It is likely that ACTH is normally more important than melanotropin in determining the amount of melanin in the skin. Here is regulation of aldosterone production. Potassium and angiotensin II increase while sodium decreases aldosterone levels. Angiotensin II increases aldosterone levels by two mechanisms. First, it induces transcription of the gene coding for aldosterone synthase. Second, angiotensin II stimulates secretion of aldosterone from the adrenal cortex. Here is a Renin-Angiotensin-Aldosterone System. In response to reduced blood pressure and reduced blood flow to kidneys, juxtaglomerular apparatus in the kidney secretes renin. Renin converts angiotensinogen to angiotensin I. In the lung, angiotensin I is converted to angiotensin II by a converting enzyme, ACE. Angiotensin II promotes production of aldosterone in the adrenal cortex through two mechanisms that I described to you earlier. Increased aldosterone level, increased salt and water retention by the kidney. As a result, blood volume increases. Angiotensin II also promotes vasoconstriction of arterioles. Both vasoconstriction and increased blood volume raise blood pressure. This slide recapitulates two functions of angiotensin II. First, it serves as a powerful vasoconstrictor. Second, it increases production of aldosterone. Aldosterone is the main regulator of sodium retention. Here is integration with pharmacology. Two drugs are used as an antihypertensive. One is lisinopril, and the other one is spironolactone. Lisinopril is an ACE inhibitor. Spironolactone is competitive inhibitor of aldosterone receptor. As a result, it decreases transcription of genes coding for sodium potassium ATPase and INA-C. Here is a clinical correlation. Hyperaldosteronism, also called Cone syndrome, has excess production of aldosterone by the adrenal glands, resulting in low renin levels. Patients with Cone syndrome have high blood pressure, muscle cramps due to low blood calcium, muscle weakness, and headaches due to hypokalemia. It is often caused by a small unilateral adrenal adenoma on either side. Treatment includes spironolactone or epinephrine. They are competitive inhibitors of aldosterone receptor. Hypoaldosteronism, on the other hand, has decreased level of aldosterone. There are two representative cases. First is Addison disease. Patients with Addison disease have low level of aldosterone and cortisol. As a result, they have hypoglycemia, hypotension, severe dehydration, and sudden penetrating pain in the legs, lower back, and abdomen. Hyposecretion of glucocorticoids leads to increased level of ACTH, which causes bronzing of skin, as shown here, compared with control. Addison disease is caused by autoimmune adrenolitis, leading to autoimmune destruction of adrenal cortex. Second case is hyporeninemic hypoaldosteronism. It is caused by renal dysfunction, leading to decreased angiotensin II. ACTH stimulation test or concentropin test is used to diagnose hypoaldosteronism. During the test, a small amount of synthetic ACTH is injected, and the amount of cortisol and aldosterone will be measured. In healthy individuals, the cortisol level should increase above 18 to 20 micrograms per deciliter within 60 minutes. In Addison disease, both the cortisol and aldosterone levels are low, and cortisol level will not rise during concentropin test. Cushing syndrome is hypersecretion of glucocorticoids by the adrenal cortex. Patients with Cushing syndrome have weight gain in the trunk and face, but not in the arms and legs. Common signs of Cushing syndrome include growth of fat pads along the collarbone, on the back of the neck, leading to buffalo hump, as shown here, compared with control. on the face, leading to moon phase, as shown here, compared with control. The most common cause of Cushing syndrome is taking corticosteroid medications. Rarely, it is caused by tumor of adrenal cortex. A related condition is Cushing disease. It is hypersecretion of glucocorticoids caused by ACTH-secreting pituitary tumor. Dexamethasone suppression test is used to diagnose Cushing syndrome. This is dexamethasone. It is a cortisol analog. A low dose of dexamethasone suppresses cortisol in individuals with normal production of cortisol. A high dose of dexamethasone exerts negative feedback on pituitary neoplastic ACTH-producing cells, as seen in Cushing disease, but not on ectopic ACTH-producing cells or adrenal adenoma, as seen in Cushing syndrome. In individuals with normal production of cortisol, a low dose of dexamethasone will lead to a decrease in cortisol levels. In patients with Cushing's disease, cortisol levels are unchanged by low doses but suppressed by high doses of dexamethasone. In patients with Cushing syndrome, cortisol levels are unchanged by low or high doses of dexamethasone. Now, let's move to part 2. We will look at catecholamines. Spherical chromaffin cells of adrenal medulla secrete epinephrine and norepinephrine. They are amine hormones stored in vesicles. These hormones prepare the body to deal with short-term stress and mediate fight or flight response. They are part of autonomic nervous system. Epinephrine acts by binding to a variety of adrenergic receptors, including alpha-1, alpha-2, beta-1, beta-2, and beta-3, and plays a variety of roles. For example, epinephrine increases lipolysis in edible tissues by activating hormone-sensitive lipase through the beta-1 receptor. It stimulates glycogenolysis in the liver and muscle through the beta-2 receptor. It stimulates glycolysis in muscle through sodium-potassium ATPase. Epinephrine triggers glucagon secretion in the pancreas through the beta receptor and inhibits insulin secretion through the alpha-2 receptor. Epinephrine also increases ACTH secretion by the pituitary gland. Together, these effects lead to increased blood glucose and fatty acids, providing substrate for energy production within cells throughout the body, especially the muscle. Norepinephrine plays a very similar role. Epinephrine promotes vasoconstriction through the alpha-1 receptor, increases cardiac output through the beta-1 receptor. Epinephrine also relaxes trachea and bronchial smooth muscle through the beta-2 receptor and dilates blood vessels for skeletal muscle and liver through the beta-2 receptor, among many other functions. Norepinephrine also plays a similar role in these processes. As we have seen before, epinephrine is derived from tyrosine in 4 steps. In the first step, tyrosine is hydroxylated to form L-dopa. This reaction is catalyzed by tyrosine hydroxylase. In the second step, L-dopa is decarboxylated to form dopamine. This reaction is catalyzed by dopa decarboxylase. In the third step, dopamine is hydroxylated to form norepinephrine. This reaction is catalyzed by dopamine beta hydroxylase. In the final step, a methyl group is transferred to norepinephrine to form epinephrine, and this reaction is catalyzed by phenylethylamine N-methyltransferase using SAM as the methyl donor. Here is a chromaffin cell of adrenal medulla. This is norepinephrine and this is epinephrine. Norepinephrine is converted to epinephrine by PNMT. It stands for phenylethylamine N-methyltransferase. Synthesis of this enzyme is increased by cortisol,